Hello students, welcome to lab number 2. We're going to be talking about molecular models. So today the lab itself is completely dry lab. There's nothing in the lab that you would previously had needed to look at. So throughout the lab manual you should be able to do every question. So the lab assignment, what you can do is basically just write down the questions and write down the answers. So all of those questions, you can write them all down and you can answer them all. Okay, so on a document, just write down all the answers, send them through the assignments, and then that should be your lab assignment for today. Okay, so. And once again, the pre-lab assignment is the questions at the pre-lab at the beginning of the chapter, and then the post-lab is your group assignment, the questions at the end of the chapter. Okay, so lab number 2, molecular molecules. Atoms are the smallest unit of matter. When you combine 2 or more atoms together, it's called a molecule, right? The result of that bonding, if you have 2 or more different atoms together in the molecule, it's called a compound, right? So a molecule itself, if you have just 2 different, or if you have 2 of the same, that's just called a molecule, right? Let's say you have a hydrogen and a hydrogen together, that's just called a molecule. But if you have 2 different, let's say you have carbon and an oxygen together, that's a compound. All of the elements, carbon is of significant importance because that's what we see in life, right? These are called organic compounds. And I would classify organic compounds as those that not only have carbon, but also have hydrogen involved too. Okay. And specifically, I would say this because carbon dioxide isn't actually an organic compound. Okay. You can't classify carbon dioxide as organic because it doesn't have hydrogen involved in it. Even though it has carbon in it, it doesn't have hydrogen in it. Those that don't contain carbon and hydrogen is inorganic. Also, diamond isn't organic. Diamond is a lattice crystalline form of carbon, but because it is so combined with so many carbons in it, it doesn't have hydrogen bonds along it, so it isn't classified as organic. Okay, the different properties which organic compounds are determined, these are called functional groups. So the functional groups that contain these compounds together, these functional groups do functions. They do work in these compounds. Okay, there's many different types of functional groups. Amino group, carboxyl group, hydroxyl group, carbonyl group. There's other types, but we'll go through these. So what is a functional group? These are groups of atoms like to bind with other molecules that contain carbon or organic molecules. These functional groups wouldn't really have a unique function or properties without, having that hydrocarbon, right? So it's like a train. If you have those trolleys at the end. Without an engine, it's not really going anywhere, right? It doesn't have steam or electric or diesel. What type of engine is it, right? But with the engine on it, that functional group, it makes it work. So the amino group is a type of functional group. Amino group has nitrogen atom and 2 hydrogen atoms. So amines, organic compounds called amines, such as amino acids. Carboxyl group, COOH. One of the oxygen is double bonded to a carbon. Carboxyl groups are found on carboxylic acids. Hydroxyl groups, this is an OH group, usually found on alcohols and sugars such as glucose. OH also binds as kind of a base, so like acid and base. OH gives bases its compound. Carbonyl group is a CO with a double bond. Don't confuse this with the carboxyl group, okay? Carbonyl group is just a C and a double bonded oxygen. Carbonyls are found in aldehydes and ketones. And these can be found in the middle of a compound depending on where it is, and it can be found at the end of a compound depending on where it is. So if we were in lab, we would be using these model kits, right? Though we can't actually build the models in lab, we can review these models on these slides. So carbon is usually black, nitrogen is usually blue, oxygen is usually red, and hydrogen is usually yellow. Let's just say that these are our colors. 6 and springs are used to form covalent bonds, right? A covalent bond is usually between a nonmetal and a nonmetal. And then long sticks are used to form carbon to carbon bonds, and springs are used to form double covalent bonds. So these springs have the double bonds between either carbon and oxygen or nitrogen and nitrogen, depending on what it is. So we have some simple organic molecules such as ethane and methane containing carbon. Well, water is inorganic because it doesn't contain the carbon-hydrogen bonds that we usually see in organic molecules. So then we say methane has an empirical formula of CH4. Right? So what is an empirical formula? So the empirical formula just means that what you see in the empirical formula is what is contained within the molecule. Right? It's a simplified form of the atoms contained within the molecule. And it's a far more complex definition within your lab manual that you can read. So methane is CH4, ethane is C2H6, and water is H2O. So if we go into ethyl alcohol, this kind of looks like a little horse, right? Because it has that bond between that OH group, that hydroxyl group, which are found in the alcohols, right? It's ethyl alcohol. So the empirical formula for ethyl alcohol, or ethanol, ethyl alcohol, ethanol, is C2H6O. Okay. An amino acid is composed of 3 groups. An amino group, so recognize the nitrogen atom right? The nitrogen atom is blue. You have the carbonyl or carboxyl group, COOH. And then the variable R group. R group is just variable depending on what the amino acid is. And the main way that you can tell if it's an amino group, right, is that nitrogen atom. And then the carboxyl group on the other end has a double bond between the carbon and the oxygen. Now, if you have alanine and glycine, these are both amino acids, right? You can see the difference. Glycine has an R group consisting of a single hydrogen atom, while alanine has an R group consisting of one carbon atom and 3 hydrogen atoms. So you should probably know the difference there. So glycine has R group with 1 hydrogen atom, and alanine has carbon atom and 3 hydrogen atoms. Otherwise, they're identical. They both have nitrogen where the amino acid is. And you can't really tell by this picture. The alanine is kind of hiding it. But the blue is gonna be, it's kind of behind that. So how do you distinguish the two, right? And I kind of just told you. Alanine has 3, so 3 times 3, alanine, that 3 carbons. And then glycine only has 2 carbons if you look at the empirical formula. Or alanine's R group is CH3, and then glycine's R group only has a single hydrogen. If you notice, these 2 amino acids, these can join. Okay, and this is in your lab manual. These can join in what's called a dipeptide bond. Dipeptide bonds form in what's known as dehydration synthesis. All right? So we know that these 2 amino acids in this polypeptide, because there's 2 nitrogen atoms, right? If there's 2 nitrogen atoms, we know that every amino acid contains 1 nitrogen atom, right? So it's a dipeptide because there's 2 nitrogen atoms in there now. So dehydration synthesis is formed when 2 molecules form together, and what comes off is water, basically. Okay. One side has an extra hydroxyl group, and one side has an extra hydrogen, right? When they bind, that hydroxyl group and that hydrogen form together to make water. Okay, so down here we have glycine that has an extra hydroxyl group in the carboxyl group, right? The alanine has an extra hydrogen in the amino group, right? So when they bind together, you have 1 ketone and then 1 nitrogen and hydrogen there. So the nitrogen and carbon bind together, right? And then you get a hydroxyl and a hydrogen floating away. Well, the hydrogen and hydroxyl aren't just going to float away. Usually they're going to bind together and make water. Dehydration just means that it's dehydrating from the dipeptide bind. It's removal of water from the large molecule that's happening. So that's a dipeptide bond. If you were to do this on the other ends, you would form another peptide bond and another peptide bond, another peptide bond. And this is how polypeptides are formed. And a polypeptide right? If you form many peptides together, that forms an amino acid chain. An amino acid chain is a protein, right? So proteins are found throughout your body. Many proteins can be enzymes or other things that do functions inside your cells. So these are how proteins are made. So other molecular molecules, we have fructose on one side and glucose on the other side. Fructose and glucose have the same exact empirical formula, right? C6H12O6. It's the same exact empirical formula, but one, fructose, has a ketone in it, and then glucose is an aldehyde, right? Fructose has a carbonyl group in it, right? Fructose is a ketone, it has a carbonyl group in it. It has that C double bonded to an oxygen. So if we look at the molecular model of fructose, we can see that carbonyl group within that chain. So it's more towards the end, right? But it's within the middle area, right? What we would call that is the 5' carbon. Okay, that's carbonyl is within the 5' carbon of fructose. Glucose has the carbonyl group at the end of the chain. It's called an aldehyde. Now, why is this important? Well, because our bodies can actually digest this differently. If you take a protein and let's say that this molecular molecule is coming through and it's breaking down a specific molecule. Well, if you take this molecule, it might be shaped differently depending on how the molecule is formed, how the atoms are formed, right? So if your protein is attaching onto it, depending on whether it's fructose or glucose, it might be shaped differently, so it might be digesting it differently. So even though the empirical formula is exactly the same, the way that this interacts with other things, let's say throughout your body, throughout other things throughout the world, it may change it drastically. And this isn't the only example. This is just sugars, right? There may be other things like complex molecules that could change this dramatically. We can do this same thing, dehydration synthesis, to form disaccharides between glucose and fructose. So glucose and fructose are called monosaccharides. These are one sugar, right? Mono meaning one, saccharide meaning sugars. Okay, so same thing between one part of the molecule containing a hydroxyl group and then the other part of the molecule containing an extra hydrogen. One hydroxyl group is removed, a single hydrogen molecule is removed, right? And then we have a carbon-carbon bond. Usually this is facilitated by some enzyme. Sometimes it's not. That hydroxyl group and the single hydrogen bond usually form into a water molecule, and then you have a disaccharide. Once again, usually at the ends of that disaccharide, you have the same type of dehydration synthesis. Okay, usually at those ends, and what you get is a polysaccharide. Okay, binding together at those other ends. Polysaccharides are what's called starches or oils or fats or other things like that. Other types of molecular models, Deoxyribose. These are nucleic molecules. Deoxyribose and ribose. So deoxyribose is a pentose sugar found in DNA, right? Deoxyribonucleic acids. These contain 5 carbons, 4 oxygen atoms, right? Comparing this to ribose, it will contain 1 less oxygen atom. It's pretty simple to tell, right? Ribose contains 5 carbons and 5 oxygen atoms. So, deoxyribose is C5H10O4, ribose is C5H10O5. Deoxyribose, right? Means 1 less oxygen than ribose. So how can you distinguish the two? You count the oxygens. So 5 oxygens, it is ribose, 4 oxygens, it's deoxyribose. Okay, and that's about it for this chapter. There are some questions that you can kind of answer at the end of this. If you want to review for the lab practical, I would suggest that before the lab practical begins. Just go ahead and you can copy these, put them on a Word document or something. This would be your review. If you have any more questions about this, please let me know. Once again, the questions for your lab manual, pre-lab questions are just the 1 through 7 in the pre-lab, right? For your lab assignment, just go through these questions. They're pretty easy, okay? Pre-lab assignment, they're pretty easy. Just go through the questions, answer them in a document and turn it in. Post-lab review questions, go through that with your group. Okay, that's going to be due at the end of the week. Okay, along with the first chapter and probably the 3rd chapter, that's going to be due at the end of the week. Okay? As for the pre-lab and the lab assignment, that's going to be due tomorrow at the end of the day. Okay? Any other questions, please email me and let me know. All right.